The eukaryotic cell. A eukaryotic cell is any cell or organism that has a defined nucleus. It has a nuclear membrane that surrounds the nucleus in which chromosomes are located. Eukaryotic cells contain organelles including mitochondria, which are cellular energy exchangers, a Golgi apparatus, which plays a secretory device role, an endoplasmic reticulum, which is a canal-like system of membranes within the cell, and lysosomes, which are a digestive apparatus within many cell types. The nucleus is a highly specialized organelle that serves as the information processing and administrative center of the cell. This organelle has two major functions. It stores the cell's DNA and it coordinates the cell's activities, which include growth, metabolism, protein synthesis, and reproduction, which takes place during cell division. The cell nucleus consists of a nuclear membrane, nucleoplasm, nucleolus, and chromosomes. The nucleoplasm is the matrix present inside the nucleus. Packed inside the nucleus is chromatin. For DNA to function, it can't be crammed into nucleus like a ball of string. Instead, it is combined with the proteins and organized into a precise, compact structure, a dense, string-like fiber called chromatin. The nucleus lets molecules in and out through gates called nuclear pores, which are strict with what molecules can enter the cell. These structures are all important to store DNA, copy DNA as needed, and make ribosomes at sites called nucleoli. The nuclear envelope is a double-layered membrane. It keeps DNA inside the nucleus and protects it from materials in the cytoplasm. The nucleus contains a nucleolus, a membraneless organelle that manufactures ribosomes. There is also a thick liquid in the nucleus called nucleoplasm, which fills the space of the nucleus. The mitochondrion is in the cytoplasm of the cell, and it is a membrane-bound organelle. It is responsible for producing ATP from chemical energy. It breaks down sugar molecules, which are used to transport energy within the cell for metabolism. Carbohydrates, fats, and other molecules in our body are sent here to be further processed to produce molecules that combine with oxygen and produce ATP. The mitochondria is red-shaped and enclosed within two membranes, called the outer membrane and the inner membrane. The membranes are made up of phospholipids and proteins, and the space between these membranes is called the intermembrane. The outer membrane protects the mitochondria and lets small molecules from up to 5,000 Daltons in and out of the mitochondria. It is completely permeable to nutrient molecules, ions, ADP, and ATP molecules. The inner membrane folds inwards, making them have infoldings called cristae to form an increased surface area for maximum cellular respiration. It contains large numbers of proteins that play an important role in producing ATP, and contains complexes of the electron transport chain and the ATP synthase complex. The inner membrane is only permeable to carbon dioxide, oxygen, and water. The matrix contains enzymes that are important for the synthesis of ATP molecules, special mitochondrial ribosomes, tRNAs, and mitochondrial DNA, which is the mitochondria's own DNA for protein synthesis. Ribosomes are found inside the mitochondria and are made up of proteins and RNA. Although it does not have a membrane-bound structure, it is considered to be a non-membrane-bound organelle due to its size and functional importance, which is synthesizing the cell's proteins. This function is also known as translation. This can happen as ribosomes are the molecular component that read information on mRNA obtained from the nucleus, and from there they can synthesize polypeptide chains for the cell. tRNA molecules are also involved as they carry the amino acids that need to be assembled to form a protein. Once protein synthesis has happened, the polypeptide chain exits through the exit tunnel of the ribosome. The endoplasmic reticulum is a network of membranes found throughout the cell and is connected to the nucleus. The membranes are slightly different from cell to cell, and a cell's function determines the size and the structure of its endoplasmic reticulum. Some cells do not need an endoplasmic reticulum, such as red blood cells, while cells that synthesize and release a lot of proteins will need a large amount of endoplasmic reticulum. Both rough and smooth endoplasmic reticulum have the same types of membranes, but they have different shapes. Rough endoplasmic reticulum looks like sheets or discs of bumpy membranes, while smooth endoplasmic reticulum looks more like tubes. The surface of the rough endoplasmic reticulum is studded with ribosomes which manufacture proteins. This gives a rough appearance, hence why we refer to this area as the rough endoplasmic reticulum. It is very important in the synthesis and packaging of proteins, and it metabolizes carbohydrates and regulates drug detoxification. The rough endoplasmic reticulum separates newly formed proteins away from the cytoplasm and transports them to another part of the cell. The smooth endoplasmic reticulum consists of tubules which are located near to the periphery of the cell. Smooth endoplasmic reticulum acts as a storage organelle. It is important in the creation and storage of lipids and steroids. The double membranes of smooth and rough endoplasmic reticulum form sacs called cistern. Protein molecules are synthesized and collected in the cisternal space, also known as the lumen. When enough proteins have been synthesized, they are collected and pinched off in vesicles. The vesicles often move to the Golgi apparatus for artisanal protein packaging and distribution. The Golgi apparatus is made of a cistern and have membrane-enclosed vesicles. The membrane surrounds an area of fluid where the complex molecules such as proteins, sugars, enzymes are stored and changed. The Golgi apparatus is composed of two regions, cis and trans. The cis phase of a Golgi apparatus is the end of the organelle where substances enter from the endoplasmic reticulum for processing, while the trans phase is where they exit in the form of a smaller detached vesicle. The Golgi complex works very closely with the endoplasmic reticulum. It receives proteins from there and chemically modifies them. Proteins, carbohydrates, phospholipids, and other molecules formed in the endoplasmic reticulum are transported to the Golgi apparatus to be chemically modified during their transition from the cis to the trans poles of the Golgi apparatus. Enzymes present in the Golgi lumen modify the carbohydrate or sugar portion of glycoproteins by adding or subtracting individual sugar monomers. In addition, the Golgi apparatus manufactures a variety of macromolecules on its own, including a variety of polysaccharides. It can then package the molecule in vesicles as storage for later use or export the molecules through the trans section where they eventually fuse with the plasma membrane of the cell. The Golgi apparatus also sorts out the wide variety of macromolecules produced by the cell and distributes them to their proper locations.